And thanks everyone for joining the webinar. Today we'd like to introduce the topic of viscosity screening for protein therapeutics, or basically how viscosity can be used as an important parameter to help scientists during the different stages of drug development. And this is our outline for today. We're going to start with the introduction of what is viscosity and what do we mean by viscosity fingerprinting, and how can we use those two concepts to help on the different stages of drug development. We will briefly review VROC technology and our family of viscometers, and then we'll move on to show some examples of applications and data that we have acquired with our instruments regarding applications such as injectability, determining the molecular size and the stability of different protein therapeutics. So we would like to start with the very basics of what is viscosity, which is defined as the resistance of a fluid to flow. A very simple way to look at it is that if you have a very low viscosity fluid, like let's say water, you will need a very small force to push it through a piston. On the other hand, if you have a very high viscosity fluid, like honey, you will need a much larger force to push that fluid through. From a rheological perspective, we like to define viscosity using an experiment that is called parallel plate shear, where we have two parallel plates. We're going to fix the bottom plate, and the upper plate we're going to move at a fixed velocity v1. Now the fluid is going to have some resistance to that movement, and to overcome that resistance we're going to have to apply a force on the upper plate. The shear stress is going to be defined as the force that we need to apply to keep that velocity v1 divided by the wet area. We're going to define the shear rate as the ratio of that velocity to the distance between those two plates. And the definition of viscosity is going to be given by the ratio of shear stress to shear rate. Now the key here is that shear rate is well defined only for a few geometries, such as cone and plate, parallel plates, circular slit, and rectangular slit geometries. So to get an idea of what viscosity is, we don't need a well-defined geometry, but to get an actual measurement of what we are going to call and refer to as true viscosity, we're going to need a geometry that is going to allow to get an accurate characterization of both shear stress and shear rate. Now, the next step is understanding what do we mean by viscosity fingerprint. It's a pretty broad concept that involves measuring the effect that any parameter, any imaginable parameter can have on viscosity. So you can think of the most basic parameters as shear rate or temperature, but you might also want to check for your formulations what's the effect that concentration has on viscosity, or what's the effect of pH, or what's the effect of the buffer type that you're using, or how is viscosity dependent on the excipient type, or any kind of denaturants that you add to your formulations, and so on, pretty much anything that you can imagine. This way, viscosity screening or viscosity fingerprinting can be used to determine drug performance in terms of injectability, molecular size in a given formulation or conditions, and stability in a given formulation or conditions. So now I would like to go through the drug development process, and it's a surprisingly long, troublesome, and expensive process that goes from the drug discovery after some basic research and determination of the disease, and that involves trying a large number of formulations, most of which are going to be discarded, and doing some preclinical trials, After that, it comes the different phases of the clinical trials, then getting the drug approved, and then finally in phase 4, where the drug is approved, but there is still some post-approval research and some monitoring of the efficiency of the drug. So if you think in terms of the early stages, you're going to have a large number of formulations, but you're going to be very limited in the amount of sample that you have. And they are also going to be pretty expensive. So you're going to have requirements such as a small sample volume and high throughput to test your samples. You're going to want to be able to get your formulations characterized using very small sample, or as small as you can. You might also want to be able to recover that sample to do some additional testing. And you're gonna want to be able to test a large number of samples. And if possible, you will want to test those in a high throughput, time efficient, and unattended manner. What you want to study is, at the end, the structure of the different active ingredients or the different proteins that you have in your formulations. When you move on into more intermediate stages, you might want to look into the application of the drug and to study things such as, for example, injectability. You're going to have a requirement of high shear to do those testings to determine the viscosity of your formulations at the shear that is relevant for your injection process, for example. In any way, during the whole process, going from the drug discovery to phase 4, after the drug has been approved, where you might want to use viscosity just as a quality control check, you're going to need a system that is able to give you an accurate, repeatable, and that can give you a measurement of viscosity in a precise and easy way. So in this presentation, what I want to show you is how our technology, VROC, the viscometer rheometer on a chip, offers solutions to many of the challenges in this process. VROC is a hybrid of microfluidics and microelectromechanical system technologies, where we basically have put together a microfluidic microchannel with a silicon pressure sensor array. VROC technology measures absolute viscosity as opposed to apparent viscosity, which is the measurement that is often offered by most viscometers. And because of the small size of the device itself, it has some advantages. The first one is that it can, or it allows for the measurement of viscosity with the smallest sample volume in the market. We can get as low as 10 microliters of sample required to do a measurement of viscosity for a single point. Another advantage is that it offers a very wide dynamic range of operation. You can measure a very wide range of viscosities, which is something that is very interesting, but you can also measure a very wide range of shear rates, going to very high shear rates, up to 1.5 million inverse seconds. Its flow-through design is going to render the high throughput that is required to test a large number of samples during the early stages of drug formulation and selection. And all this is achieved in a fast, accurate, and repeatable way with an accuracy of 2% of the reading and a repeatability of 0.5% of reading. Here are some of the details of how the technology actually works. We're going to be pumping a fluid through the microfluidic channel at a known flow rate. Since we know the dimensions of the channel, we're going to be able to establish the shear rate. Now, the pressure sensor array at the bottom of the channel is going to characterize the pressure as a function of position, and it's going to give you the pressure drop. The pressure drop is going to be proportional to the shear stress. And as we mentioned before, the ratio of these two, shear stress and shear rate, is going to give you the definition of viscosity. Powered by VROC technology, RIOCEN offers two levels of laboratory viscometer rheometers, the MVROC and the microvisc. The MVROC is the most versatile viscometer in the market. It can measure viscosity with as little as 20 microliters of sample. The shear rate range goes from 0.5 inverse seconds to 1.4 million inverse seconds, and the viscosity range goes from 0.2 to 200,000 centipoise. It allows for temperature control, and it allows to set shear and temperature sweeps in an automatic way. Of course, it's powered by VROC technology, so it offers the superior accuracy and repeatability that our customers are used to. Our other instrument, microvisc, is portable. As a result, it requires a slightly larger amount of sample to make a single measurement of viscosity, and it has a somewhat limited range in terms of shear rate range, viscosity range, and temperature range. However, as I mentioned, it's portable, and one of the things that I like most about microvisc is that it offers disposable pipettes. Last month, during our last webinar, we were very excited to present our newest product, VROC, which is a high throughput, fully automated viscometer rheometer. Among other things, VROC features 10 microliter sample volume requirement for a single measurement. It's compatible with a 40 vial rack or a 96 volt plate. It allows you to set shear rate and temperature sweeps. It has a built-in Peltier temperature control that controls temperature between 4 and 70 degrees during testing, and a secondary temperature control that controls the sample rack temperature between 4 and 40 degrees. As I just mentioned, VROC offers the smallest sample volume requirement for a measurement of viscosity in the market. And in the figure on the left, you can see a proof of the concept. You can measure the viscosity of a fluid at a given temperature in a really accurate way with just 10 microliters of volume. Well, if that's not enough for you, VROC also features sample retrieval. What that means is that you're going to be able to test your sample, make it flow through the VROC cell, but then once you have your measurement, you're going to be able to retrieve it back into the syringe, and then you have two options. One is to recover your sample, and the other one is to test it again at a different shear rate or at a different temperature, and that's going to allow you to do studies of shear stability and thermal stability. Going back to the range of operation, VROC technology offers a really wide dynamic range of measurement, and that's achieved by combination of 4 different types of flow cells that vary the membrane stiffness. With that, we have chips that are classified as A, B, C, and E, and we combine those with different channel depths ranging from 20 to 300 microns. The wide dynamic range of operation that allows us to achieve goes from 0.2 centipoise to 200,000 centipoise in terms of viscosity, and in terms of shear rate, we can measure from 0.5 inverse seconds to 1.5 million inverse seconds. That wide dynamic range, the ease of use, and the precision of our instruments have allowed us to work with a large number of applications. Those range from industrial applications such as flow coating and mixing, screen printing, and so on, to biopharmaceutical applications such as protein stability or injectability of protein therapeutics. All right, at this moment, I would like to move on into the different applications that we're going to discuss during this presentation. I will start with injectability. I will move into some application that is known as intrinsic viscosity, which allows us to measure the molecular size. And I will move on to some stability results that we have achieved with our instruments. So we have offered in the past a webinar regarding injectability, and if you're interested in getting more details about this specific application, you can check those through our website. However, here I'm going to give some brief idea of what it is and how our instruments can help you achieve your goals in this regard. The idea is very simple. Biopharmaceutical companies are trying to reduce the frequency of dosing. That's usually achieved by increasing the concentration of the active compounds or the proteins in the different formulations. That increasing concentration is going to lead to a higher viscosity. Now, on the figure on the right, you can see some very interesting behaviors. The first one, as I just mentioned, is that the increasing concentration for different proteins is going to lead to an almost exponential increase on viscosity. The second one is that the different proteins are going to have very different behavior. So you need a system that is able to characterize this in an accurate way. Additionally, and that you can observe in this figure, is the fact that as you go to higher concentration, you might have a dependence of viscosity on shear rate or non-Newtonian therapeutic. And you're going to need to characterize those three different phenomena to be able to establish injectability, which is the force required to inject a specific drug at a given injection rate. Next, I would like to show some examples of how this actually works. The first one, in the first table in the slide, you can see an example for Newtonian therapeutics. We have used two different injection rates, which give us two different shear rates, and with the same viscosity, because this is a Newtonian therapeutic, we have estimated the injection force. When we compare those to the report injection force that has been measured in an actual injection device, we can see that there is an exceptional agreement and that the contribution of piston friction is almost negligible. Next, when we add more complex flow, or we have a non-Newtonian therapeutic, what we look at is how the viscosity decreases as we increase the shear rate. That is commonly characterized by the power law exponent. As we decrease that n, which is the power law exponent, we're increasing the non-Newtonian behavior of our sample. So our sample will more strongly shear thin. That's gonna have several effects. One of them is that the shear rate at the wall is gonna be larger than you will expect for a Newtonian therapeutic. So as you decrease n, you will get a larger shear rate. The other effect is that that will further reduce the viscosity. So you're going to have lower viscosity than what you will expect for a Newtonian therapeutic, and that's directly going to affect the force. So that as we show in this example, for different non-Newtonian fluids, you can get a significant decrease in the force required to inject non-Newtonian therapeutics. And the assumption of Newtonian behavior or measuring viscosity at low shear rate might lead you to reject some of those formulations that with a more accurate characterization at high shear rate might be perfectly applicable for your application. Next application that I want to discuss is intrinsic viscosity. Intrinsic viscosity is a measurement of a solid's contribution to the viscosity of the solution. As you add solid molecules to a given solvent or solution, you're going to get small increases in viscosity. That put in a mathematical way give us the definition of intrinsic viscosity, which is the limit when the concentration of those solid molecules goes to zero of the relative increase in viscosity divided by the concentration. Now, intrinsic viscosity, it's a powerful and well-known tool to determine both molecular size and molecular weight for polymers. And it can be used as well for biopolymers to determine those two properties. To determine the molecular weight, we can use the Mark-Hoehn-Sakurada expression, which relates intrinsic viscosity with a power law to the molecular weight, where K and A are the Mark-Hoehn-Sakurada parameters, which are available for a large number of polymer and biopolymers in different solvent combinations. Now, if we know the molecular weight, we can use the Einstein-Simha expression to obtain the hydrodynamic radius. The hydrodynamic radius is a representation of the solid molecule size in that specific solvent or environment. It can be used to test conformation, stability, structure, and denaturation and melting temperature in the case of protein molecules. As a result, intrinsic viscosity can be used as a complementary technique to DLS to determine molecular size, with two key advantages. One is that as we are measuring a bulk property, it's not so sensitive to local impurities. And the second one is that we are not limited by the bulk viscosity, and we don't need to dilute some of our formulations. Here I'm just showing a couple of examples of measurements of intrinsic viscosity for biopolymers such as BSA and gamma globulin. This is how you actually measure intrinsic viscosity. You create several formulations in increasing concentrations, and you measure the viscosity, and then you're going to extrapolate to zero concentration to obtain the value of intrinsic viscosity. That is going to allow you to establish molecular size and structure, stability, and conformational changes triggered by pH, temperature, and so on. As you can see, in the case of BSAs, we have available molecular weight, so we could estimate the hydrodynamic radius, and we obtain values that are in really good agreement with the literature reference. Next, I want to move on into examples of viscosity screening or viscosity fingerprinting. The first one that I want to show is how the viscosity is affected by concentration and formulation. So in the figure on the left, you can see different concentrations of BSA in PBS at different shear rates. In this case, concentrations are relatively low, and you can see how all solutions behave in a Newtonian way. You can see how viscosity increases with concentration, as you will expect. On the figure on the right, you can look at somewhat more interesting behavior. You can see how viscosity increases with concentration for BSA and PBS, but you can see how the viscosity versus concentration profile changes depending on the buffer solution that we use. If we use water, we observe a slightly higher increase in viscosity with concentration. If we change the protein that we're studying, you observe a much more significant increase on viscosity, the viscosity profile with concentration. Now, all these measurements showed in these two figures are representations of average of 3 repeats, and they are all performed with 100 microliters of each of the formulations. What that means is that you can measure more than 20 measurements at different shear rates with a single 100 microliter syringe using our technology. All right, the next example that I want to discuss it regards denaturation or temperature stability. On the figure on the left, you can see how the viscosity of the same formulation of gamma globulin changes as a function of time depending on the incubation temperature. You can see how for 40 degrees there is no changes in viscosity, and how for 60 degrees there is a significant increase and a further significant increase for 64 degrees. Now, that can be very interesting to study the temperature stability of different formulation, but what you can see is that you can characterize not only the dependence versus time of viscosity, but you can characterize how the shear thinning behavior depends on that incubation temperature. So you can see how for 40 degrees during 3 hours, on the figure on the right, we still have a perfectly Newtonian fluid. At 60 degrees, we start to observe a decrease in viscosity with shear rate. And at 64 degrees, this is a slightly higher temperature due to the protein unfolding. In 20 minutes, we observe a very significant shear thinning behavior. The next examples go to show what's the effect of adding an excipient to our formulation. In this case, we have added sucrose to our gamma globulin and PBS solution of 100 milligrams per ml. We're doing all these measurements at the same shear rate, and as you can see, the blue signs, which are the same that I showed in the previous figure, show a significant increase in viscosity versus time. Our control solution, which is just PBS with sucrose, has a constant viscosity independent on time, as you will expect, but the solution with the excipient, with the sucrose on it, has a much less effect in terms of viscosity increase with time at a given temperature. That's because the protein itself is being stabilized due to the addition of the excipient. Next example is a check on the effect of a denaturant concentration on the viscosity of a solution. Here we have BSA solution, 100 milligrams per ml in PBS, and we add urea. As you can see, after some critical concentration, we see protein unfolding with a exponential increase in viscosity after that concentration. You can also see how for our control solution, PBS and urea, on the figure on the right, we get really good agreement with literature data based on capillary gas measurements. With that, I would like to summarize how today we have presented that high throughput screening of viscosity can be a very useful tool within the different stages of drug development for the characterization of protein therapeutics or protein formulations. And that can be used in applications such as injectability of those protein therapeutics, intrinsic viscosity to determine hydrodynamic radius and molecular size and structure. It can be used to test the stability of proteins under different conditions. That can be temperature, it can be pH, it can be different excipients, it can be different denaturants, and you can also check for the reaction kinetics of those reactions, of those degradation or denaturation reactions. With that, I would like to thank everyone for joining the webinar, and we'll take some questions.